Thanks, folks. Thanks for having me. Thanks for sticking with us during a bunch of lectures today. I'm going to just jump right in. So I got a great introduction, but I like to put this up and say I went to school a couple of places, worked a couple of places, thank my mom for making me do my homework, and just kind of go in and jump in and say that there are a couple of companies I'm going to mention that I either work for or with, and I will highlight those as I go. Good science piece of the talk. This is what I do now. This is what I work in. I work in genetic engineering, I work in DNA sequencing, and systems biology, which is where kind of big data meets DNA. And we're trying to apply all those things, especially to personalized health. And this is what I want to talk to you about today. I want to talk to you about the world economy and how I see biotechnology as being about 1/3 of this entire world economy. And that sounds like a big chunk until you think, well, that's all of medicine. That's all of agriculture. And then more and more of manufacturing, materials, energy. And then it doesn't sound like such a big chunk. So I think this is a highly relevant subject for our forum today. And I also tend to see the world as sort of surrounded by this nice little loop of DNA because that's what we're working with today. We're working with digitized biology. These are the pieces I want to talk to you about, and we're calling it here DNA sequencing, genetic engineering, and democratization. Really, we probably should have called it reading, writing, and hacking DNA, because that's basically what we're doing. And to jump in, DNA sequencing is something, and it's really kind of the reason I get to talk to you today. I was very privileged to work on a team that designed next-generation DNA sequencing. We came up with something that didn't work, and then it finally did, and people paid us a lot of money for it. And I found out that those stock options were not just little pieces of paper, and that was wonderful. The way it works is you start with a little glass slide. It's got some channels in it. You pipe in some DNA. The DNA sort of ends up on the slide like a kelp forest there. And then we rebuild each strand of DNA with specially marked chemicals so that it shows up under a microscope. And we go ahead and move this microscope around and take a new picture every 100 microns or so and look at this and go ahead and do this over and over and over again and build up a picture of what somebody's DNA sequence looks like. And we were able to do it in such a way that we're getting it cheaper and cheaper all the time. And this is one of those nice logarithmic graphs that we're in love with at Singularity University. And so what looks like a nice general linear curve is really a Moore's law. This is getting cheap fast. And to give you an idea, back in 2001, we finished the Human Genome Project using old last-generation technology, and that first project cost about $2.7 billion. Well, you can buy a B-2 bomber for $2 billion, so you'd have enough left over to pay for Tumblr whenever you did it. Then we got better and better at it. In fact, James Watson, a company called 454, sequenced his genome, and it cost about $1 million to do that by 2007. $1 million will get you a nice rundown castle in, upstate New York, maybe Long Island, where Jim lives, or apparently about an 800 square foot 2-bedroom in the upper mid, I don't know, but it's looking kind of rough. But by this time last year, we were able to do a whole genome, and it cost about as much as a good, reliable Toyota, a used one, and this is from East Palo Alto from an ad that I clipped out. The air conditioner doesn't work, little hubcap mismatch problem, but it'll get you where you're going. And we're not showing any signs of slowing down on being able to do this. If you look at this graph and you sort of project out, even very conservatively, this is what the next maybe 10 years looks like. So actually, this year, in January of this year, we hit my prediction that I had made 8 years ago. Human genome for under $1,000, and that's a nice, relevant number for medicine. It's something that makes a human genome really less than the cost of a chest x-ray. So that's nice. 2 more years, we're going to be able to do the same thing for the cost of a delivery pizza. And then just a little bit after that, in 2020, it's going to cost less to sequence a human genome than it costs to flush a toilet. And I always like to say, also, whenever we flush one of those toilets, we're going to go ahead and analyze everything in there. There'll be a little genome sequencer sitting there. I don't know, maybe that worries you, but it's probably going to be good for your health. You may not want to be ordering a lot of delivery pizzas. You'll be doing the other thing. But for sure, the marginal cost of a human genome by 2020, I think, is going to be about a penny. And that really opens up a new world for us and some of the things that we can do. I wanted to talk a little bit about some of those implications here from sort of a financial perspective and a forecasting perspective. What we're really seeing is that biology is now falling under Moore's Law. And when I say falling under, I mean it's subject to the dictates of Moore's Law. Things are getting cheaper all the time, but really it's falling far under. We're beating Moore's Law. We're beating Moore's Law with a stick. The progress that we're seeing, especially in DNA sequencing, we're going at about a 5x a year change in throughput and efficiency every year, again, driven by the same economic forces in Moore's Law. And that's got kind of an interesting effect on the economics of doing this, because we're one of the few industries that's actually held back by microprocessor improvements. We're like, could you guys go ahead and hurry and catch up? And the way that this works is you see the Moore's Law line there in blue, and then our DNA sequencing line in green, where we're making this cheaper and smaller and faster, using less expensive chemical reagents. And really, they've crossed so that while the pie is getting smaller and smaller, a larger proportion of that pie is being taken up by the electronics, the data storage, the processing and analysis cost. And really, the story over the next 10 years is going to be, how do we do that and do it well and get it to doctors and people and health systems? The other piece, and this I downloaded this morning on a plane on the way here, to look at what the latest figures are published by the government for the cost of a genome. This lags reality by about 9 to 18 months because they take this out of published papers. But there are two interesting pieces of this graph. You can see the Moore's Law piece, but you can see the inflection where things get a lot cheaper than Moore's Law. And then you can see the part where it's kind of evened off a little at the end. The company that's really responsible for both of those is Illumina. And anybody who follows this industry knows a little bit about them. But that's where the next generation sequencing technology was perfected, and they've dominated the market to so much of an extent that they've got nice, large cash reserves, and they go ahead and buy up all of the new pieces in the field. And anybody who's got a new development, they take that, productize it, send it out, or sometimes, wait a little bit to see what's going to happen. And there's a little bit of an effect where they are protecting their turf and maintaining that margin, although that's ripe for further disruption as the next pieces come online, what we are calling next generation sequencing, because we're good at sequencing and not so good at making up names for it. So you may not believe me about this 2020, it's going to be a penny, and I want to kind of emphasize a couple of things. I'm talking about marginal cost, that it costs so much to do 1 million genomes. The million and first genome is just going to be another penny, and that's good to know. And it's not so unbelievable when you think about we're shifting all this stuff down to the size of a little bitty nano machine on a highly engineered chip, and we're using about a single drop, just a few nanoliters of expensive reagent, and we're going to process these things through at speed for whole populations. The other thing is we're really seeing a difference in how we're doing this, and people understand this when you talk about computers. If I told you you could go in and buy an ENIAC for a penny, you would know, well, no, you couldn't do that, but you can get several times the processing power of an ENIAC for a penny. And you can get a decent laptop for about $800, and you can go ahead and get a bunch of time online with Amazon's EC2 cloud to process things for, anywhere between $100 and $10,000, depending on what you're doing. It's very elastic. We're seeing the same thing. We're seeing sort of a market segmentation in all of these different pieces we're doing. And these are kind of my little forecasting figures I like to look at: a single human genome, what it costs to do genomes in bulk, what it costs to do an exome, which is the sort of medically relevant portion of the genome for now, what it costs to do a gene scan, like 23andMe is a company, and that's available to consumers now. And we'll see if it still is, see what the FDA says over about the next year or so. So this is kind of where I'm going with these costs here. The big effect that I see this happening is that in the very near future, medicine shifts much more from being a reactive piece where, you get sick and somebody tries to do something about it, to more predictive, where we're able to look at, for instance, cancer risk and what's going on there, where we're able to look at what diseases somebody is likely to get over the course of their life by doing a DNA profile maybe on the day they're born, and then going ahead and setting up appropriately rigorous screening as they go through so that we stop trouble before it happens. And I was challenged on this. I was talking to a large insurance company, and they said, we hear about new high-tech pieces all the time, and you guys are always selling us the moon, and it costs a lot of money, and what we see is that it costs a lot of money. We don't get the returns. And I was able to actually come back with two really nice recent examples. And these are all pieces that kind of develop out of this field of pharmacogenomics, where we're actually looking and seeing how we can predict what drugs work and don't work for people by looking at their individual genetic makeup, that actually really kind of pays for a gene scan and now increasingly a gene sequence, especially if somebody's older, takes multiple drugs, was taking some high-cost drug, or some drug that has a high cost to themselves personally if it doesn't work or doesn't work right. We're also seeing big strides in rare disease, studying longevity, but especially cancer. Cancer is a genomic disease, and we're finding all sorts of new ways to scan it, treat it, do new things about it. And I could talk to you about that for the rest of the afternoon. Let me just move on and give a couple of examples here. Here is a current product that is out now that's genomically based, and it replaces amniocentesis. So this is something, and I was able to use this to talk to my insurance company and say, you know, you guys worry about things costing more or less. Well, here's something, amniocentesis, a couple of technicians, a doctor in a room, some expensive ultrasound equipment, versus you just take a regular blood sample on an outpatient visit even from an expectant mother. You're able to pick the free DNA out of her blood that belongs to the fetus and look at that. There's no risk to the fetus. It's something where we're seeing accuracy that's on par with what we get from amniocentesis, and we're actually now able to look at more and more things with this. Another thing that we're starting to see is early detection of cancer by doing the same sort of technology, taking a blood sample from an individual and looking for DNA that's floating around in the blood. And with some confidence, I'm going to predict that by 2020, we're not going to be taking mammograms. We're not going to be doing colonoscopies anymore. And I'm glad, and it was kind of hard to find slides that weren't too... You'll notice I have cartoons up there. But that's indicative of how, you know, whenever my sweetie goes and gets a mammogram, she's like, I just went to the crusher. And I'm like, oh, that doesn't sound good. And I want to, you know, make sure I'm not saying anybody, you know, stop doing all your mammograms and colonoscopies. But whenever these new tests come online, I think it's going to end up, we're going to look and say, man, the x-rays that we're having to go through, the discomfort, it's not worth it. And people are not doing the test the way they should. But if you go ahead and give a little blood every time you see your doctor, or, you know, even like some kind of quarterly checkup where it's not going to be a doctor visit, you're going to head off a lot of problems. And you're not going to wait until a tumor is so big that you can see it. That's going to seem very primitive. Whenever tumors are less than the size of a pinhead, it's really nice to do some sort of prophylactic chemotherapy treatment and knowing which chemotherapy would work for that particular kind of tumor. And not even having to know where it is in your body, just saying, here we go. So we're getting to that point, and I'm already seeing people, papers, standard care procedures where they're saying, let's go ahead and start using these technologies instead of some of the existing tests. I want to mention really quickly, we're even seeing, as the sequencing power improves for these technologies, things like uBiome, which is a company that tests the DNA not of you, but of all the bacteria that colonize you. And that gets really interesting, and I want to talk about that in a little bit, because wouldn't you like to know kind of what's living on you, and do you need to do something about it? Wouldn't you like to know how that affects your health? Turns out that we carry 2 pounds of bacteria, maybe 2.5 pounds in our gut, and that's about the same weight as the human brain, and it may have some effect on your mood. It does definitely on your digestion, on your ability to withstand different environmental changes. So we're learning more about sort of all these cells that we carry around. This is all good. This is all interesting. And then it gets even better whenever we start factoring in the next generation machines that come up. And we're seeing companies that are really challenging, you know, using a brand new technologies that may even steepen that curve. We're seeing places like Nanopore, Oxford Nanopore, with a handheld sequencer that actually plugs into a USB port. And I'm pretty sure if I give this same talk at the end of the decade, I'm going to walk in with something about this size, wave it around, collect your DNA, and be able to tell, different people, not everything, I won't do a full sequence, but I'll do enough where I could build up a picture of your face on the screen and get an idea of what you look like and maybe what you suffer with. Like, look for this guy who walks with a limp and is about this tall. We'll be there pretty quick. And that's the start. What gets really interesting is, after we can read everything, what happens when we can start writing things and changing it. And really, I've been amazed, I've been really impressed. We're entering an era where we are at the renaissance of genetic engineering. All sorts of amazing, interesting things are going on. I want to just hit on a couple of them fairly quickly. One is we're starting to see drag and drop genetic engineering, where you don't have to know much about what you're doing. You say, I want to start with this compound, I want to produce this, I want to program some bacteria to do that and make it happen. And this is what that looks like. We're able to use basically DNA and write our own DNA to do software development in living organisms, and especially in E. coli. And this is very interesting. E. coli, ideal conditions, every 20 minutes, they double. And so you can start with one, and as Peter has showed us, at less than the end of the day, you have over 1 billion little minions making things that you want them to make. Whenever we engineer these bacteria, we start with a CAD tool, but we're not drawing a house. We're going ahead and drawing out the DNA that we want to use. This is a free tool. This is something that high school students download and start playing with. Whenever they get a nice design they want, and here we were splicing jellyfish glowing genes into a nice little E. coli. They go ahead and cut and paste that sequence onto a website like DNA 2.0, down the road from me back in California, hit add to cart, go ahead and put in a credit card number. For about 20 cents a base, they get some DNA shipped out to them in 72 hours, printed to order. Reconstitute it, use it to treat a bacteria, and then see if the thing that you wanted it to do works or not, and then go ahead and go back and iterate again on an engineering cycle, unlike what we've seen before in life sciences. It's not as tight as doing web page development, but it's a lot more satisfying. This is where we're at now. And this is really driving a revolution, something people like to call synthetic biology. I'm not in love with the term synthetic biology. I think it's a poor marketing term. I think it suggests some things that make people a little bit queasy whenever you start thinking, especially about the artificiality that's put in. And anytime you talk about synthetic something, if you say synthetic leather, that suggests something that's inferior to normal leather. It's not something that you want. Unless, perhaps, you're a cow, then synthetic leather sounds pretty good. And I wanted to highlight a company that we work with at Singularity University, these guys at Modern Meadow. And what they're doing is they're applying large-scale tissue culture techniques, basically, to first going to grow leather and then grow hamburger, grow steaks. And it's kind of funny, people started out calling them synthetic biology. They don't really like that. They started talking about this being a 3D bioprinting. They don't like that. It kind of goes, and they're narrowing down the term, but what we're going to eventually just call this is food. And we're going to do this in such a way where the environmental impact is much lower. You would not believe how many gallons and gallons of water it takes to go ahead and grow a cow up to maturity, to then cut away most of its body in a pretty gross process. And you don't even have to be a vegetarian looking at it saying, I don't think that this is moral. It's just wasteful. It's bad engineering. So being able to do this where you really control it and have good food security. And as the rest of the world comes online and wants more and more meat, more of a Western diet, really this becomes world-saving technology. We're starting to see this, and I like to call it industrial rather than synthetic biology, because really it's the industrial application of microbes that live all around us. We've been doing this for a long time. In medicine, we've been doing this for at least 30 years. We've got here a pile of pig pancreases about as tall as I am to make 1 liter of insulin. And that's how we used to do that up through the 70s. Now, we had a nice breakthrough where they were able to program, as really the first modern biotechnology company, E. coli to make the 2 parts of insulin and put it together. And that was back in '78. And you can tell by looking at the sideburns in this picture. It's been a while. We've known how to do this. We're getting better and better at it. We're trying to scale up and do tanker cars full of product. And a decent example here is what I like to think of as green solar panel cells. These are nice little plastic bags that have genetically engineered algae floating in them that are busily churning away and excreting a precursor to a biofuel. So this isn't, corn ethanol, with a lot of price supports and harm to the environment. This is a really nice way to make a drop-in fuel easily and cheaply. But it's hard to do. It's hard to scale. Where we're seeing the most success in this industrial biology are companies, and a lot of these companies that were making tanker cars or trying to full of biofuels are now scaling down to smaller chemicals, boutique chemicals, things in the food flavoring industries, things that are nutraceuticals. A great example is making vanilla. If we actually look at how vanilla and taste and smell is made, about a tenth of it can be met by growing natural vanilla in the world, would strip all those plants out. So we're actually now doing a lot of this by using microbes to grow a vanilla substitute. And that's a $300 million a year industry. And there are a lot of little industries like that that are sort of ripe for the picking that some of these companies are starting to target. As we go through this, a good question to ask is not just what are we going to do industrially with microbes, but what are we going to do with ourselves? How long until we are actually genetically engineering humans? And I hope you won't be surprised to know that, well, that's here. We are actually doing it. We've kind of been doing it with some genetic selection with IVF for about 10 or 15 years. We have it down with IVF whenever a blastocyst, a little, not a baby, not a fetus, but a baby fetus, is at about the 8-cell stage. You can pick one of those cells and just take it out and look at it. And it doesn't hurt the blastocyst. The blastocyst goes on and develops. It doesn't, come missing an arm or a leg. You get a full, nice, healthy human. But you can go ahead and analyze that cell and say, does this kid have cystic fibrosis? Is this kid a carrier for cystic fibrosis? Because if we're looking, let's go ahead and make sure we're picking the best of the fetuses we're going to look at. And more, we're actually kind of going on that slippery slope to not just preventing disease, but improvement. Whenever you've got a human being at the start of life, and you can do something with these 8 cells, you can not just look, not just read, but you can write. And so we're starting to see that now. We actually have not just this prenatal genetic diagnosis, but mitochondrial therapy, where we have children now with 3 parents genetically. We're starting to see improvements where we can go ahead and alter this genetic code as we go through. Some of this is a little spooky, and it's coming really fast. Here, I just want to point out, here are 3 nuts and bolts technologies that support genetic engineering, and especially genetic engineering in mammals and humans. And this is sort of the base cost for doing an experiment, for making something to see if it changes a gene the way you want it to, starting with zinc finger proteins, moving down through talins, and then CRISPR. CRISPR is an acronym to remember because it has made it nice and cheap and easy. And to give you an idea how fast this happens, how this is coming, this is when we discovered CRISPRs. We discovered the mechanism existing in a bacteria in 2010. And in fairly short order, we applied this and used it to genetically engineer microbes, then plants, then animals. Then we did the first human tissue experiments. We've had unconfirmed reports of humans being engineered with this, at least fetuses, coming out of China. And already we've got a company that is using this to work on human disease, and they're in a patent fight. And so that's what we can do in 48 months. Very fast, very quick, and this is going to have a lot of impact. CRISPR is something you're going to hear more about. It's going to be the nuts and bolts that drives, I think, human genetic engineering, therapeutically and for enhancement. The last thing I kind of want to say here, this is a little bit of a thought piece, is about the microbial nation. We've got all these microbes that live in us and on us, and they're going to stop being wild and start being domesticated. We're not going to have strange things that give us diseases. We're going to have things that work for us, that help tame our environment, that help us do the things we need to do. In the little bit of time that I've got left, I want to sort of jump out and talk about how we are democratizing this technology, and a little bit about how we're doing cheaper tools. And just give you a couple of examples really fast. Any laboratory has a centrifuge. Now you can go ahead and do the same thing by 3D printing a Dremel fuge, putting the little plastic piece on a Dremel tool. Bzzz, and there you've got a nice field-capable, cheap, easy-to-use centrifuge, and it's cheaper even than what you get off of eBay. We've got 3D printed pipetters. This is under $100. It uses a straw as the disposable element. It uses a spring from a Pez dispenser to do the return on the thumb press. So this is nice and easy and cheap. Here's a copy machine for DNA that started, this would be a $8,000 machine. These guys sell this for $599. And it's got some great uses. And we're seeing all these things being used in garage labs. Skip through that. And here's one of my favorite garage labs, the first one I visited out in Silicon Valley, tissue engineering lab, nicer than the one at the $5 billion company I worked at the time. And I was looking at doing some garage experiments, and several other people were. We said, let's put all of our stuff together, and we came up with this idea for a thing called BioCurious. BioCurious is a hackerspace for biotech. We did it out in Sunnyvale in California, put a bunch of stuff in one room, a lab out in a little commercial park. For $100 a month, people can come and access this space. It's all nonprofit. It's kind of to encourage open access to these tools so more people can use them. But to give you an idea, if you want to see what it looks like, you can go to New York City's own biohacker lab, GenSpace. If you're down on Wall Street, hop on the number 2, take the 3rd stop in Brooklyn, and get off, and then go over on Flatbush Avenue, and you can visit them. Give them a call first. It's an open lab, but they prefer to have an appointment rather than drop-ins. But it's a really nice space, and it was one of the very first ones in the world. To give you an idea of what we do at these places, this is what the lab in Sunnyvale looks like. We teach a lot of classes. Here we see some students who are developing a DNA assay by first building their own PCR machines, their own copy machines for DNA. The next day they come back, they extracted, purified, prepared their own DNA for sequencing. Then they sequenced it. One of these young women was looking at her BRCA1 gene, the gene that determines high risk for breast cancer, ovarian cancer. This is what she found, that she was clear and didn't have a problem. We do a lot of mentoring of scientists, the next generation. Here's the little girl who's actually helping teach a class in genetic engineering. She was a 2nd grader, 8-year-old at the time. She's now teaching more classes for us. And we are working with inventors and entrepreneurs. This is the big thing, the big impact I think that comes from this. How can you get to hello world in biotech? And what we're really seeing is by dropping the entry costs, by going ahead and making an outsourcing ecosystem, using some of the citizen science that exists out there, using different services that are available, and then not taking IP, we're really encouraging new people to come in the field. We're seeing the rise of biotech garage inventors doing really good work. Already we've got 50 companies, more than that have come out of BioCurious, millions of venture capital that have been invested, a few really great successes, and we're seeing hackerspaces like this worldwide. This is a little map. Brooklyn has the martini glass. Here are a couple of spaces that I've visited just in the last year going around the world, and these are guys who established, and were already doing this before BioCurious was established, wanting to do it, and we were able to kind of lead the way, and more places are calling us up saying, how can we avoid problems, what can we do? This is really the center of innovation coming up. So we're trying to bring on a scientific revolution. We're trying to get more tools into more hands. We think that that gives us more breakthroughs. And I just want to end, I was just in Denmark looking at a couple of new biohacker labs and talking to some engineers there. And I got to visit with folks and tour facilities, 2 really great companies, and said, well, the future of biotech really looks less like Novo Nordisk with all the good old, this is how we do regulatory, this is how we handle money, this is how we do marketing, and more like Lego, where it's small pieces, very precisely defined, that people can use to build, without having a lot of existing knowledge themselves, new solutions and test and iterate very quickly. So I'd like you to think about that. I'd like to leave you with that. And thank you for your attention, and thanks for listening.